Another oral abstract yesterday was about complex karyotype and the role of it in the GAIA trial. So to remind everybody, the GAIA trial is this very large 4-arm study where FCR or bendamustine rituximab was compared to either venetoclax-rituximab or venetoclax-obinutuzumab or a triple combination, venetoclax, ibrutinib and obinutuzumab. And this was part of the intergroup study with the Germans, the Nordic group and our Hoven group. And we did now a biomarker side analysis where we looked to complex karyotype. And I think the relevance here is that we know that complex karyotype has an adverse events, but we still don't really much use it in the clinic because of difficulties, but also because of difficulties in interpretation, because most of the times p53 aberrations are a confounder in those studies. So in this study, patients with a del17p or a p53 mutation which was centrally collected and examined were excluded from the study. So we now have a group complex karyotype without p53 and del17p. And to quickly go to the results, so first of all we saw that 3 or more aberrations is a poor prognostic marker for chemoimmunotherapy, but not for venetoclax-based combinations. In that case, 5 or more aberrations are important, and that we saw also with a multivariate analysis. The other thing we saw is that translocations are a poor prognostic marker for both groups of patients, so both chemoimmunotherapy and venetoclax-based treatment. We saw also that came back in overall survival. And another important thing is a clonal drift. So what we did is we collected samples at baseline and we collected patients after relapse. And what you see is that the number of patients with a complex karyotype very much increased after chemoimmunotherapy, but not after venetoclax-based treatments. We saw that the number of aberrations increased after chemoimmunotherapy and not after venetoclax-based treatments, and also the number of patients that developed a del17p increased significantly more in patients after chemoimmunotherapy than after the targeted treatments.